So this video is the continuation of the last lecture on the development of the embryo till the second week. So in the last video we saw that how this blastocyst gets embedded within the endometrium. So in this video we will see what happens further to the blastocyst, how it gets embedded, and how it peeps through the endometrial layer and gets attached with the inner lining of the endometrium. So as I already said, on the 6th day implantation occurs. So during the consequent days, that is 7th, 8th, 9th day, it gets through the endometrium. As a whole, this blastocyst gets through the endometrial layer. It passes through the compact layer, the spongy layer, and it gets attached with the basal layer of the endometrium. So during 7th, 8th and 9th day, it gets through the endometrial layer, and just during the 10th and 11th day, it gets embedded within the basal layer like this. It gets embedded within the basal layer of the endometrium. So during 10th and 11th day, it gets deeply embedded. In the endometrium. So once the blastocyst enters into the endometrium, the pathway through which the blastocyst enters is sealed by a fibrin plaque. Initially it is sealed by a fibrin plaque. So after that, around 12th or 13th day, a new mucosal layer develops. This is the mucosal layer. It develops around 12th or 13th day. So now just trust me that a second week of development can be compacted under the rule of 2. What is that rule of 2? I hope by this time you all might know what is a trophoblast, the outer cellular layer of a blastocyst. So during the second week of development, the trophoblast develops into 2 layers. One is cytotrophoblast and the other is syncytiotrophoblast. Then the embryoblast, the inner cellular mass that develops into 2 germ cell layers, which is called epiblast and hypoblast. And third is 2 cavity develops during the second week of embryonic development. Those 2 cavities are amniotic cavity, and the second is the yolk sac, primary yolk sac. Then it develops into a secondary yolk sac, and finally it transforms into a chorionic cavity. Then the extra embryonic mesoderm. They are also two in number. One is splanchnopluric mesoderm and the second is somatopluric mesoderm. So we already discussed what are all the things that happens during the 8th, 9th, 10th and 11th day in the endometrium. So now we are here to discuss about what happens to the blastocyst from the 8th day onwards. We have discussed what happens to the blastocyst. What are all the changes that takes place in the blastocyst. What happens to the blastocyst on 8th day of development? So this is the trophoblast, which is the outer cell mass of the blastocyst. So this is the embryoblast, which is the inner cell mass. So the pole at which the embryoblast is attached to the trophoblast is called the embryonic pole. And the other pole is called as abembryonic pole. I'm just removing the embryoblast. That means we are now going to focus only on the changes that happens in the trophoblast. What are all the changes that happens during the 8th day of the embryonic development to the trophoblast? So during the early embryonic development, the trophoblast is just a single layer. So as the time passes, it develops into two layers. So as the time passes, the trophoblastic cells develops into two layers of cells. The inner layer, you can observe in this picture that in this diagram that the inner layer is properly arranged and each of the cells has a clear-cut cell membrane and the nucleus are properly and regularly arranged. Whereas the outer layer, the cytoplasm is confluent, that it is not packed within the cell membrane and it is not definite in shape or anything. So the nucleus are interconnected with each other like a syncytium. As these cells are derived from the trophoblast, they are called multiple nuclei plus derived from trophoblast. So they are called as syncytiotrophoblast. The inner layer, each and every cell are having a clear-cut definite shape and cellular membrane and they have their own nucleus, so they are called cyto means cell. The cells are properly arranged and they have their definite characteristics what must a cell have, so they are called as cyto. Since they are derived from the trophoblast, they are called as cytotrophoblast. So during this transformation from single layer to double layered, the mitotic figures appear on the cytotrophoblast. So thus the cells in the cytotrophoblast are rapidly multiplying and giving rise to new cells. And the new cells move outward from the cytotrophoblast. They lose their membrane and thus their protoplasm fuses together. So they appear as like a syncytium. So from this it is clear that the syncytiotrophoblast is derived from the cytotrophoblast. Another important thing is that the syncytiotrophoblast develops more on the embryonic pole than on the abembryonic pole. So around 9th day, the syncytiotrophoblast develops some cavities, develops some fluid filled cavities like this. These fluid filled cavities are also called as lacuna. So multiple cavities starts developing in the syncytiotrophoblast around 9th day. So then around 9th or 10th day, these cavities starts merging with each other. So therefore forming a large cavity. So as I already said, the blastocyst is now embedded within the endometrium. And this is the embryoblast. So around 9th day the lacuna developed, and around the 9th and 10th days of development the lacuna fuses together. So what happens, the spiral arteries within the endometrium just start supplying the lacuna. So the blood starts flowing into the lacuna. So now the maternal blood has entered the blastocyst. So this circulates throughout the blastocyst. Now the maternal blood circulates throughout the blastocyst through the lacunar spaces that are found in the syncytial trophoblast of the blastocyst. So somewhere the blastocyst is connected with the arterial blood supply, and somewhere the venous drainage, right? So the arterial blood supply will be the high pressure system, and the venous blood drainage will be the low pressure system. So the blood flows from the arterial end to the venous end. So this is the initial establishment of uteroplacental circulation. This initial establishment of uteroplacental circulation occurs by about 11th and 12th day of embryonic development. So what happens sometimes by the 13th day, the fibrin plug must be formed. Once the blastocyst enters into the endometrial lining, the fibrin plug must be formed to seal the opening through which the blastocyst enters into the endometrium. But if it does not form properly, so what happens, the maternal blood enters the blastocyst and circulates through the lacunar spaces. There is a chance of the maternal blood to bleed through this improperly sealed fibrin plug, that is the improperly sealed endometrial lining. There is a chance of bleeding through the improperly sealed endometrial lining. So what a pregnant lady may think that she is not conceived yet anymore. There is a chance of thinking like that when the bleeding occurs at about 13th day or 14th day of the embryonic development. I'm not saying 100% that it will occur. I'm just saying that it may occur if the fibrin plug does not form properly. If this fibrin plug does not seal the endometrial lining through which the blastocyst enter, if it does not form properly, then there is a chance of bleeding to occur. So again, let us come to the cytotrophoblast. So this is consider this as the cytotrophoblast, right? These are the cells of the cytotrophoblast, and this is the syncytial trophoblast, and there are some lacunar spaces around. What happens around the 14th day? Some of the cytotrophoblastic cells make columns of the cells like this. They start appearing like this in between the syncytium and the lacunar spaces. They start appearing and developing like this. So these are called the primary villi. Of course, the lacunar spaces are filled with the blood and they are also interconnected. Of course, it is right. But from around 14th day, some cells of the cytotrophoblast just starts developing like this and start making columns like this in between the syncytial trophoblast, and they are called as the primary villi. So I hope I discussed everything that is related with the trophoblast and the two layers that it emerges from the trophoblast, that is the cytotrophoblast and syncytial trophoblast. Let us now concentrate about the embryoblast. What is the development that occurs in the embryoblast during these days, right? So now let us concentrate on about what happens to the embryoblastic cells. Embryoblast. Of course, consider this as the cytotrophoblast. And this is the syncytial trophoblast. Of course, there are some lacunae. This is during the early part, that is when the trophoblast just splits into the cytotrophoblast and syncytial trophoblast. What happens to the embryoblast during the time at which the trophoblast just divides into two layers, that is a cytotrophoblast and syncytial trophoblast. And that's what I didn't mention about the villi, primary villi. the syncytial trophoblast. Of course, the lacunae are filled with the maternal blood. Right, like this. So what happens? Some of the cells of the embryoblast are pushed, they are opposed towards the embryonic pole. Some of the cells of the embryoblast are opposed towards the embryonic pole. So they are called as amnioblast. There is a reason for they being called as amnioblast. Of course, there is a reason. There develops a cavity by leaving some of the embryoblast to become the floor of that cavity. These embryoblastic cells are columnar in shape. This cavity is called amniotic cavity. Since these amnioblasts form the roof of the amniotic cavity, they are called as amnioblast cells. Another group of cells lie below the this layer of cells. This layer of cells is called as epiblast. It forms the floor of the amniotic cavity. And another group of cells, they are cuboidal in shape and they lie just below the epiblastic layer of cells, and they are called hypoplastic cells. And they form the roof of the another cavity, which is called as primary yolk sac, which is developed from blastosphere. So what happens now? The cells of the hypoblast just starts proliferating and seals the cavity of the yolk sac. So now trust me that this is the amnioblast cells. I hope by now you know that these cells line the amniotic cavity and it forms the roof of the amniotic cavity, and this cell layer is columnar cells and it is called as epiblast, and it forms the floor of the amniotic cavity. And this cell layer is a hypoplastic cell layer which is cuboidal in shape and forms the roof of the primary yolk sac, which is derived from the cavity of the blastocyst called the blastosphere. And this proliferates to line the cavity of the yolk sac, primary yolk sac. So around the 9th and 10th day, the yolk sac starts secreting a connective tissue layer and it extends all around the yolk sac and it extends till the amniotic cavity. It surrounds the amniotic cavity too. And it is called as the extra embryonic mesoderm, since it lies outside of the embryo. So like this, the yolk sac starts secreting a layer around the primary yolk sac and also the amniotic cavity. So this layer of cells is called as extra embryonic mesoderm. Soon after, the extra embryonic mesoderm just starts splitting into two layers. So like this, leaving the gap in between the two layers. Before explaining what are these two layers, I would like to draw the cytotrophoblast and syncytial trophoblast. This layer is the cytotrophoblast, and above that it is the syncytiotrophoblast. And of course there are some fluid filled spaces. Now it is filled with maternal blood and it is called as the lacunae. So as I already said that the primary yolk sac starts secreting a connective tissue layer, and it is called as extra embryonic mesoderm. Soon it just starts splitting into two layers, leaving a gap in between. So the visceral layer, that is the inner layer which is derived from the extra embryonic mesoderm, it is called as splanchnopluric mesoderm. Splanchnopluric mesoderm. And the outer layer which is attached to the cytotrophoblast, it is called as somatopluric mesoderm. So of course it happens around the 14th day, so the chorionic villi is now developed. You know why sometimes something makes us interesting. I'll explain what is that. Even after the development of extra embryonic mesoderm and a splitting of the extra embryonic mesoderm into splanchnopluric mesoderm and somatopluric mesoderm, some part of the bilaminar germ disc is still intact with the cytotrophoblastic cells, and this part of bilaminar germ disc which is still intact with the cytotrophoblastic cells is called as connecting stalk. This is the structure which later on develops into an umbilical cord. So after some time, some part of the primary yolk sac just detaches from the cavity and falls into the space between the splanchnopluric mesoderm and somatopluric mesoderm. Right. So now the yolk sac gets transformed as secondary yolk sac. So now the extra embryonic coelom, which is the space between the splanchnopluric mesoderm and somatopluric mesoderm, is now called, once the primary yolk sac detaches from the cavity of the yolk sac and falls into the extra embryonic coelom, now it gets transformed into chorionic cavity. So by the end of the second week of development, it is proven that the true baby comes out of the bilaminar germ disc. Of course, it will be then transformed into trilaminar germ disc by a specialized process, of course. But by the end of the second week, we can literally confirm that the true baby comes out of the bilaminar germ disc. So another important thing I would like to mention is about the chorionic plate. The extra embryonic somatopluric mesoderm, the cytotrophoblast outer to that layer of the somatopluric mesoderm, and the syncytiotrophoblast which is still outer to that of the cytotrophoblast, together, all these together form the chorionic plate. So the extra embryonic mesoderm, the cytotrophoblast and the syncytiotrophoblast, all these together forms the chorionic plate, and later on just only one part of the chorionic plate rapidly develops into placenta. I hope I discussed each and every point that happens during the development till the second week of embryonic development. So I'd like to revise it rapidly according to the rule of 2. So first of all, trophoblast. So trophoblast just splits into two layers, that is the cytotrophoblast and syncytiotrophoblast. So again, after some time, that is the splitting of the trophoblast just happens during the 8th day. After some time means just around the 9th day, the syncytiotrophoblast starts developing multiple cavities. Those cavities are called lacunae. So during after being embedded within the endometrium, Some of the spiral arteries give branches to the lacunae so that the maternal blood just starts circulating within the lacunar spaces of the blastocyst. So what happens? The maternal blood just travels all through the blastocyst and it just starts supplying the blastocyst, and it is the what to say that the initiation of the uteroplacental circulation, which happens during the 11th and 12th day of the intrauterine life. Of course, at the same time, the embryoblast just splits into the epiblast and the hypoblast. The epiblastic cells are columnar in structure and the hypoblast are cuboidal in structure. The epiblastic cells forms the floor of the amniotic cavity, and it forms the roof of the hypoblast forms the roof of the yolk sac, primary yolk sac, which is derived from the blastocyst. So, of course, later on it will be converted into trilaminar germ disc. Now it is bilaminar germ disc. So it will be converted into a trilaminar germ disc by the process of gastrulation. That is something different that happens after the second week of development. So till the second week, it is more than enough to know about the epiblast and the hypoblast. And some of the cells, of course, the embryoblastic cells, some of the cells are pushed or opposed towards the embryonic pole, so they form the roof of the cavity. Those cells are called as the amnioblast. Then to speak about the cavity between the amnioblast and the epiblast, there forms a cavity. So this cavity is called as the amniotic cavity, and blastocele, it is derived from the primary yolk sac. So then it, after a part of the primary yolk sac being detached from the yolk sac cavity, it falls into the extra embryonic coelom, so that the yolk sac is converted into the secondary yolk sac. And once this primary yolk sac, part of primary yolk sac falls into the extra embryonic mesoderm, it gets converted into the chorionic cavity. Then extra embryonic mesoderm, to speak about that, so the yolk sac starts secreting some connective tissues that spreads all over the outer part of the yolk sac, the cavities, the yolk sac and the amniotic cavity. So except one place, that is the place where the connecting stalk remains. Except that place, the extra embryonic mesoderm just aligns all those cavities, that is the yolk sac and the amniotic cavity. And after some times, it also splits into two layers, that is the splachnopluric mesoderm, which is closer to that of the yolk sac and the amniotic cavity, and the somatopluric mesoderm, which is attached with the cytotrophoblast.